Welcome to the deep dive. Today, we're cutting through the noise to get you well informed on one of your body's most intricate and vital systems, how your blood maintains its perfect balance.
Yeah, it really is.
We're talking about blood plasma proteins, how your body stops bleeding, and then crucially, how it cleans up the mess afterwards.
It's truly a marvel of biological engineering.
Mm.
Think of it as your body's internal highway system, its rapid response repair crew, and its waste management team all rolled into one.
Mm.
We'll uncover how blood plasma is far more than just a liquid. It's a dynamic, living system that keeps everything flowing, fixes itself on the fly, and defends against threats.
Our mission today is pretty straightforward. We want to distill the most important nuggets of knowledge from a detailed biochemistry chapter.
Mhm.
We're going to guide you through the key concepts, the biochemical pathways, and even some real-world clinical examples, helping you visualize these microscopic processes without needing a textbook right in front of you.
Right, making it accessible.
Consider this your express ticket to understanding the essential chemistry of your blood.
Let's do it.
So let's dive into that liquid part of your blood, the plasma.
Mm.
Beyond the red blood cells carrying oxygen, what's actually in there doing all this heavy lifting?
Well, plasma is essentially the non-cellular fraction of your blood. It's
Right.
mostly water, yeah, but it's also packed with nutrients, hormones, electrolytes, and crucially, a really rich array of proteins.
Okay.
These plasma proteins are truly biological multitaskers, each with several essential roles that keep your body in balance.
So not just a simple watery fluid then. What's the first big job these proteins tackle?
One of their most critical functions is regulating fluid distribution. Imagine your capillaries, those tiny blood vessels weaving through every tissue.
Okay.
At one end, your heart's pumping pressure, what we call hydrostatic pressure, pushes fluid out of the blood and into the surrounding tissues. But you don't want all that fluid just staying there, obviously. So at the other end of the capillary, it's the osmotic pressure exerted by these plasma proteins, particularly a protein called albumin that acts like a gentle magnet.
Pulling it back.
Exactly, drawing that fluid back into the bloodstream.
Sure.
This elegant balance prevents swelling in your tissues, keeping your fluid levels perfectly tuned.
That's a fascinating bit of choreography, these proteins pulling water back where it belongs. And I know proteins are also the ultimate transporters in the body. Is that true for plasma too?
Absolutely.
Mm.
Many vital substances, things like lipids and steroid hormones, they aren't water-soluble.
Right. They need help.
They can't just dissolve and travel freely through your blood.
Yeah.
This is where plasma proteins, especially albumin, step in as their personal transport vehicles.
Okay, albumin again?
Yeah. Albumin is your body's major serum protein, making up about 60% of all the protein in your plasma.
Wow, 60%.
And despite its relatively small size, it's responsible for a huge 70% to 80% of plasma's osmotic pressure.
So this one protein, albumin, is not only a fluid regulator, but also a universal carrier. What exactly does it ferry around?
Oh, it's quite the workhorse. Albumin carries everything from free fatty acids and essential minerals like calcium and zinc to steroid hormones and bilirubin.
Okay.
And what's fascinating is how many common drugs also bind to albumin.
That's interesting.
Yeah, this binding can significantly impact how effective a drug is and how long it stays active in your system, which is a major consideration in medicine.
So if albumin is so crucial, what happens if someone doesn't have enough? Is it catastrophic?
Well, what stands out here is the body's incredible adaptability. In rare conditions like analbuminemia, where individuals essentially lack albumin,
Right.
they surprisingly often experience only moderate edema or swelling.
Really? Just moderate?
Yeah, because other plasma proteins can actually step up and partially compensate for albumin's absence, taking on some of its fluid regulating and transport responsibilities. It's a testament to the body's backup systems.
Amazing. So these proteins keep our fluid balanced and transport essential molecules. What about defending against invaders? Are they involved in our immune system?
Definitely. Plasma proteins are right there on the front lines of your immune system. You have immunoglobulins, which you probably know better as antibodies.
It's antibodies, right?
These are specialized proteins secreted by plasma cells, and their job is to seek out and bind to specific foreign invaders, what we call antigens, to neutralize threats like viruses or bacteria.
And the complement system sounds like it's a squad of proteins that works alongside these antibodies.
Precisely. The complement system is a complex network of about 20 proteins that gets activated in a cascade, like a series of dominoes falling.
Okay.
This activation can happen when they bind to those antibody-antigen complexes, or even directly to certain bacterial components. Once activated, they generate biologically active peptides that trigger inflammation, attract other immune cells to the site of infection, and help clear away harmful complexes.
So they clean up and call for backup.
Kind of like that, yeah. Think of them as the precise cleanup crew that also calls in reinforcements.
So they clear threats, but inflammation itself can cause damage if it's not controlled. Does the blood plasma have a way to sort of put the brakes on that?
Oh, absolutely. What's fascinating here is that the body has built-in controls. Another group of plasma proteins, called protease inhibitors or serpins, are crucial for managing inflammation.
Serpins.
Proteins like AHA1 antiproteinase, also known as AHA1 antitrypsin or AAP,
Right. Yeah.
AAP, limit the destructive potential of enzymes called proteases. These proteases are released by activated immune cells, like neutrophils, during an immune response. The serpins form complexes with these proteases, essentially trapping them and preventing them from damaging healthy tissues. It's damage control.
I recall a really striking clinical example tied to AHA1 antitrypsin. Here's where it gets really interesting for me.
What a great connection. Yeah, a common point mutation in the AAP gene can lead to greatly diminished secretion of this protein from the liver. Individuals with this condition are at a significantly increased risk of developing emphysema.
Wow.
Without enough AAP in the lungs to neutralize elastase and other proteases, these enzymes continuously break down lung tissue, leading to irreversible damage.
And smoking makes it worse.
And here's the kicker. Smoking can dramatically worsen this by oxidizing a critical amino acid in AAP, MET358, which completely destroys its ability to bind to and inhibit proteases.
So genetics and lifestyle, a perfect storm.
Exactly. Creates a perfect storm for lung damage.
Okay, so plasma proteins are busy keeping us hydrated, healthy, and protected from internal threats. But what happens when things go wrong, when a blood vessel is damaged and there's a leak? How does your body prevent bleeding out?
That's where hemostasis comes in. This is your body's emergency repair system, the physiological response that stops bleeding.
Hemostasis. Got it.
When a vessel is cut, a blood clot, or thrombus, forms through a process called thrombosis. It's a rapid, localized response involving two key players: tiny cell fragments called platelets, and a complex series of chemical reactions known as the coagulation cascade.
So platelets are like your body's tiny patch kit, the first responders.
Exactly that. Platelets are not full cells, but non-nucleated fragments butted off from giant cells called megakaryocytes in your bone marrow.
Megakaryocytes, right?
Their primary role is to form immediate mechanical plugs at injury sites. They also secrete regulators that kickstart the full clotting process and aid in vascular repair.
So they patch things up constantly.
Think of the constant minor wear and tear on your capillaries. Platelets are quietly patching these up daily without you even noticing.
How do these platelets actually spring into action? Do they just sort of glom together?
It's far more sophisticated than that. Non-activated platelets have a complex internal membrane system, which gives them a huge surface area for clotting reactions. But upon activation by an injury, they dramatically change shape, extruding long, sticky arms called pseudopods. They also release substances from internal granules that are absolutely crucial for clotting.
What's a real-world scenario where these tiny patch kits aren't working right?
A classic example is idiopathic thrombocytopenic purpura, ITP.
ITP.
This is an autoimmune disease where your body produces antibodies that mistakenly attack specific glycoproteins on the surface of your platelets.
Oh, so your own body attacks them.
Yeah. The spleen then recognizes these marked platelets and removes them from circulation. With fewer platelets available to patch those constant minor damages to your tiny capillaries, one early sign you might notice is small red spots on the skin called petechial hemorrhages.
Tiny leaks.
Essentially tiny unpatched leaks.
So once activated, how do platelets form that initial plug?
Platelet activation involves three fundamental steps: adhesion, aggregation, and secretion.
Okay, adhesion first.
First, adhesion. Platelets stick to the exposed subendothelial layer of the damaged vessel, primarily to collagen, and a large protein called von Willebrand factor, or VWF.
VWF.
Specific glycoproteins on the platelet surface, like GPIA and GPI, are key to this initial sticky connection.
And then they start sticking to each other, which is aggregation.
That's right. This initial adhesion triggers a cascade within the platelet, leading to the release of compounds like ADP from its granules. ADP is a potent platelet activator itself.
Okay.
And it unmasks more binding sites on the platelet surface, specifically GPI by tier. These sites are essential for fibrinogen to bind, which then acts like a molecular glue, linking platelets to each other and forming a platelet aggregate.
The initial soft plug.
Exactly, that initial soft plug. Thrombin, a key player we'll discuss soon, also strongly activates platelets.
You mentioned von Willebrand factor, VWF. How important is that in this whole process?
Oh, VWF is absolutely critical. It's a large glycoprotein that not only helps platelets adhere to the injury site, but also acts as a chaperone, stabilizing factor VIII.
Factor VIII?
Which is a crucial protein in the deeper clotting cascade, protecting it from degradation. The importance of VWF is highlighted by VWF deficiency, which is actually the most common inherited bleeding disorder.
Most common. Wow.
Yeah. It impairs both platelet adherence and, indirectly, the full clotting cascade due to low factor VIII levels.
So issues with VWF can cause bleeding, but there are also problems with the platelets themselves that prevent them from responding to VWF, right?
Precisely. For example, Bernard-Soulier syndrome is a bleeding disorder caused by mutations in that GPI protein on platelets.
The sticky protein.
Yeah, preventing them from adhering to VWF at the injury site. Then there's thrombotic thrombocytopenic purpura, TTP, a dangerous condition characterized by tiny clots forming inappropriately throughout the circulation.
That sounds bad.
It is. This often involves defects in the VWF-specific metalloprotease, an enzyme that normally regulates VWF's size. If this enzyme isn't working, abnormally large VWF multicolmers can form, which are too effective at binding platelets, leading to excessive and dangerous clotting.
It sounds like a beautifully orchestrated immediate response. But that soft clot made of platelets and fibrinogen isn't strong enough on its own, is it? It needs to be strengthened.
Absolutely. This is where the blood coagulation cascade steps in, transforming that initial plug into a stable, durable seal.
Cascade.
It's a complex, amplified series of reactions, primarily involving proteins that are initially inactive precursors or zymogens. Think of them as sleeping giants.
Ooh.
They become active enzymes when they're precisely cut by the preceding enzyme in the cascade. This sequential activation creates a rapid, localized burst of clot formation, building upon the initial platelet plug.
So these are like biochemical dominoes falling, each one activating the next. What are the main types of players involved?
You got it. First, we have the proenzymes, which are primarily serine proteases. These include factors like 7, IX, X, and prothrombin.
Lots of factors.
When activated, for example, factor IX becomes IX, they literally cleave and activate the next proenzyme in line. Then there are cofactor proteins like tissue factor, factors V and 8.
Right, the helpers.
They don't have enzyme activity themselves, but act as crucial binding sites, helping to assemble these enzyme complexes and accelerate the reactions. Finally, we have regulatory proteins like protein S and protein C, which act as the off switches to prevent overclotting.
I remember hearing about extrinsic and intrinsic pathways in relation to clotting. Are those distinctions still relevant?
You know, historically, those terms describe pathways triggered by external trauma, like exposed tissue factor, extrinsic, or internal stimuli, intrinsic.
Okay.
But what we now understand is that these pathways overlap considerably and work in concert. They both funnel down to a central point, activating factor X.
This hurt 10.
Exactly. This entire process relies heavily on activated platelet membranes exposing negatively charged phospholipids, which provide a crucial platform or scaffold for these reactions to happen intensely and locally.
So factor X is a big deal, but what's the ultimate goal, the master enzyme this whole cascade is trying to produce?
The ultimate goal is to activate thrombin from its precursor, prothrombin. Thrombin is a powerful serine protease, the master conductor of the clotting process, if you will.
The conductor, I like that.
Its activation is incredibly accelerated and localized on the platelet surface. Factors like prothrombin and factor X have special sites called carboxyglutamate residues.
Gamma-carboxyglutamate.
Right, which were formed in your liver in a vitamin K-dependent process. These sites bind calcium, which then anchors these factors precisely to the negatively charged platelet surface. This complex assembly accelerates thrombin formation by tens of thousands of times.
Wow, that's huge acceleration.
Ensuring that intense clotting activity happens exactly where it's needed and nowhere else.
Thrombin sounds like it's not just a product, but also a catalyst that speeds up its own production.
Indeed, thrombin plays a remarkable dual role. It not only converts fibrinogen into fibrin monomers, which then spontaneously link together into that initial soft clot, but it also dramatically amplifies its own formation.
How does it do that?
It does this by activating cofactors V, 8, and 11, essentially supercharging the cascade. It also gives a further boost to platelet activation. It's a positive feedback loop initially,
So that soft fibrin clot isn't quite strong enough for a lasting repair, right? How does it get strengthened into something more stable?
Exactly. The initial fibrin clot is fragile. Thrombin also activates factor 13 to factor 13.
Factor 13.
Factor 13 is unique among the cascade enzymes. It's the only one that's not a serine protease.
Oh, interesting.
Instead, it catalyzes a transamidation reaction, which essentially acts like a molecular welder, creating strong covalent cross-links between the fibrin monomers.
Welding them together.
Pretty much. This transforms the soft clot into a robust, three-dimensional hard clot that can withstand mechanical forces and enzymatic breakdown. This sturdy fibrin network then traps aggregated platelets and other blood cells, forming a complete and stable plug at the injury site.
You mentioned that some of these clotting factors have special carboxyglutamate residues formed in a vitamin K-dependent process. Why is vitamin K so crucial here?
This raises a really important point for understanding both your body's chemistry and how certain medications work. Vitamin K is absolutely essential.
Okay.
It's required for the modification, the decarboxylation, of several key clotting factors, specifically factors 2, 7, IX, X, and protein C and S, a process that happens in your liver.
Right.
Without this specific chemical modification, these factors cannot bind calcium, and without calcium, they can't properly attach to the negatively charged platelet membranes to form those critical activated complexes. This severely impairs the entire coagulation process.
So if someone is deficient in vitamin K or a drug interferes with it, their blood won't clot properly. This sounds like the mechanism behind common anticoagulant medication.
Precisely. This is why warfarin, also known as Coumadin, is such an effective anticoagulant and is widely used to prevent dangerous blood clots.
Coumadin, right.
It's a vitamin K antagonist, meaning it's structurally similar to vitamin K, and it competitively inhibits the enzymes your body needs to regenerate active vitamin K. This prevents the necessary go-carboxylation, leading to the production of non-functional clotting factors.
So they're made, but they don't work right.
Exactly. Without these properly modified factors, your blood's ability to clot is significantly reduced.
Okay, so we've got this amazing amplified system for building a strong clot. But what's to stop it from just clawing everything inside you?
Right.
And once the vessel is healed, how does that clot disappear?
That's the absolutely critical next step. Clotting is incredibly tightly regulated through both feedback amplification and equally powerful inhibition mechanism.
Right, the brakes.
Exactly, the brakes. And then there's a completely separate system designed to gracefully break down the clot once its job is done, a process called fibrinolysis.
Fibrinolysis.
The genius here isn't just that it clots, but its elegant feedback loops. A self-amplifying system that starts precisely where needed, then has built-in brakes to prevent clotting everywhere else.
Thrombin, the master conductor, seems to be involved in everything. Does it also play a role in regulating itself?
It does, which is a fantastic example of the body's sophisticated balance. Thrombin itself has both prothrombotic or clot-promoting and antithrombotic or clot-inhibiting roles.
Dual roles again?
Yeah. While it dramatically amplifies its own formation and stimulates platelet aggregation at the injury site, it also binds to thrombomodulin on healthy endothelial cells. That's the lining of your blood vessels.
Okay.
This binding actually abolishes thrombin's clotting function, and instead allows it to activate protein C, a key off switch for the entire system.
So protein C and protein S are like the body's natural anticoagulants stepping in to stop the process.
Exactly. Activated protein C, APC, in a complex with its cofactor protein S, acts like a molecular shredder, destroying the active cofactors, factor eta and Vol.
Shuts them down.
Shuts them down. This dramatically decreases thrombin production, effectively putting the brakes on the clotting cascade. APC also stimulates the release of prostaglandin I2, PGI2, which further reduces platelet aggregation.
And deficiencies here are serious.
Oh yes. If we connect this to clinical reality, deficiencies in protein C or S significantly increase the risk of dangerous venous thromboembolism. Tragically, individuals homozygous for these mutations often don't survive infancy without immediate replacement therapy.
And there's a well-known genetic mutation that affects this process, making people more prone to clots, isn't there?
There is, and it's quite prevalent. Factor V Leiden.
Factor V Leiden, right.
It's a common genetic mutation that makes factor V resistant to being cleaved or turned off by APC.
So the off switch doesn't work on it.
Exactly. It means the off switch can't effectively disarm factor V, leading to its prolonged activity and a higher risk of deep vein thrombosis. It's one of the most prevalent genetic risk factors for familial clotting disorders in people of European descent.
What other sophisticated mechanisms does the body have to control all these powerful proteases circulating in your blood?
Well, your plasma contains a group of serine protease inhibitors, the serpins, which we mentioned earlier. They make up about 10% of plasma proteins.
Right, the Protease police.
Ah, yeah, good way to put it. They act as traps for protease involved in coagulation, controlling unwanted activity. A key example is antithrombin III, AT3, which specifically and irreversibly inactivates thrombin.
H3.
It's one of the body's most important defense mechanisms against widespread clotting.
I recall heparin being related to this, a common anticoagulant drug. How does that fit in?
What's truly fascinating here is how heparin works. Heparin, a complex sugar molecule called a glycosaminoglycan, dramatically enhances AD3's action.
Enhances it. How?
It binds to AD3, changing its shape to make it thousands of times more effective at interacting with and inactivating thrombin. Heparin acts catalytically, meaning it's released and reused, making it a very efficient accelerator of thrombin inactivation.
So it supercharges the inhibitor.
Precisely. This elegant biochemical partnership is directly exploited in medicine. Unfractionated and low molecular weight forms of heparin are widely used as fast-acting anticoagulants.
And are there newer drugs that are even more specific in how they prevent clots?
Yes, the field has advanced significantly. For instance, fondaparinux is a synthetic molecule that specifically enhances AT3's interaction with factor XAM, inhibiting it without affecting thrombin or platelet activity.
More targeted.
Exactly.
Yeah.
This specific targeting helps avoid complications like heparin-induced thrombocytopenia, HIT, and unexpected clotting and platelet reduction that can sometimes occur with traditional heparin.
Okay.
We also have direct thrombin inhibitors, like bilirubin,
So yeah.
which was actually inspired by molecules found in leeches, nature's original anticoagulants.
Leeches, really?
Yeah. These agents bind directly to thrombin, making them more targeted. And for preventing platelet aggregation, drugs like clopidogrel, a P2Y12 receptor inhibitor, directly block the ADP receptor on platelets, stopping them from clumping together. These newer, more specific drugs often offer advantages in safety and efficacy.
So the body has all these intricate internal mechanisms, but what about the blood vessel itself? Does the healthy lining contribute to preventing unwanted clots?
Absolutely.
Yeah.
Your healthy blood vessel lining, the endothelium, is naturally anti-clotting or non-thrombogenic.
Non-thrombotic.
Endothelial cells are negatively charged, which helps to repel your negatively charged platelets. They synthesize anti-aggregating vasodilators like PGI2 and nitric oxide, which keep blood flowing smoothly and discourage platelet activation.
Mm-hmm.
They also produce thrombomodulin and heparin sulfate, which, as we discussed, enhance thrombin inactivation. The intact endothelium is your body's constant guardian against inappropriate clot formation.
So once a clot has successfully formed and the vessel is healed, how does your body get rid of it and clear the path?
That's where fibrinolysis comes, in the elegant process of dissolving the clot.
Fibrinolysis breaking fibrin.
Exactly. This involves the degradation of fibrin in the clot by a serine protease called plasmin. Plasmin is formed from its inactive precursor, plasminogen, by plasminogen activators like tissue plasminogen activator, TPA, and urokinase, UPA.
TBA and UPA.
Now, plasminogen has a high affinity for fibrin, which is key. This ensures that plasmin is generated and active right at the clot surface, where it's protected from circulating inhibitors like A2 antiplasmin.
So it works locally.
Yes, allows it to efficiently break down the clot without degrading healthy circulating fibrinogen elsewhere in the blood. Very targeted breakdown.
And TPA is actually used in emergency medicine, right? To break up clots?
Yes, it's a prime example of applying this biochemical knowledge to save lives. Plasminogen activators like TPA are used therapeutically to dissolve pathological clots in critical conditions, such as a myocardial infarction, a heart attack, or an acute stroke caused by a blood clot in the brain.
Wow.
It's a powerful tool to restore blood flow when time is of the essence.
What stands out to you most about how all these complex systems interact in a real patient?
You know, what's truly striking is the delicate balance and interconnectedness. Take Peter K. from our chapter's waiting room, a 6-month-old with unexplained bruising and swollen knees.
Right, the case study.
His diagnosis of hemophilia A points directly to a deficiency in factor VIII. This isn't just a missing protein. It's a profound breakdown in a crucial amplification step for factor X activation, impairing the entire coagulation cascade.
Because factor VIII is needed to boost factor X.
Exactly. If factor VIII can't effectively accelerate factor X's activation, then thrombin production is severely limited, and a stable, hard clot simply can't form properly. In the past, this was a life-threatening condition. and treatment with factor VIII concentrates carried significant risks, like viral transmission.
Hmm.
Today, thanks to biochemical understanding, recombinant factor VIII is available, making treatment much safer and highlighting how scientific insight directly translates into profound clinical advancements.
So what does this all mean for us, the listener? What's the big takeaway?
It means that from maintaining your body's fluid balance to preventing both excessive bleeding and dangerous clotting, your blood plasma proteins are constantly working in an invisible, synchronized concert.
A constant balancing act.
Truly. Any disruption, whether it's a genetic mutation in a factor, an autoimmune attack on platelets, or the interference of a drug, can have profound systemic effects on your health. Understanding these biochemical pathways isn't just academic. It's fundamental to diagnosing and treating a wide range of diseases, giving us insight into both vulnerability and resilience within our own bodies.
What an incredible deep dive into the microscopic world within our veins. We've seen how plasma proteins maintain fluid balance and immunity, how platelets and a cascade of enzymes come together to staunch a wound, and how these processes are meticulously regulated to keep you safe and healthy. It's really quite amazing.
Indeed. It's an elegant testament to the body's complexity. Think about how a tiny chemical change, a single amino acid substitution, or the availability of just one vitamin like vitamin K can have such profound effects on something as fundamental as blood.
Yeah. It literally dictates the difference between life-threatening bleeding and dangerous clotting. It really makes you appreciate the biochemical symphony constantly playing out inside you, perfectly balancing creation and destruction.
Absolutely. Thank you for joining us on this deep dive into the essential biochemistry of blood. We hope you feel a little more well-informed and perhaps even more curious about the amazing processes happening within you right now.
Thanks for listening.
Until next time, keep exploring.